Hi everyone, my name is Annette and I will be presenting my REU research with Dr. Fu about histidine tautomer identification using carbon-13 and nitrogen-15 solid-state NMR spectroscopy. So proteins are one of the 4 main macromolecules that make up your body, and histidine is one of the 20 amino acids that make up proteins. And it's very important in protein conformation or protein folding, which is the structure and what the functions of proteins is dependent on. An example of why histidine is important is it can be used to create histamine, which is the main chemical your immune system uses in allergic reactions, as you've probably heard of antihistamine. So there are 3 different versions or tautomers of histidine shown here, and it's important to differentiate between them because though they are very similar, they bond with different amino acids differently, which can affect protein folding, and they are all present in equilibrium in biological states. They're very pH sensitive, so the sample that we are looking at has a pH of 6.3, and we'll see how that affects which tautomers are present. And the technique we're using is solid state NMR. So these are the radio frequency pulse programs we're using. This is carbon-carbon correlation, so showing the connectivity between the carbons, and this is carbon-nitrogen. And here the carbon signal is filtered through the nitrogen signal, so we will only see peaks from carbons which are connected to nitrogen. So here is the data that we collected. The x-axis is the carbon peaks and the y-axis is the nitrogen. And you can tell by the number of peaks how many tautomers are present. So there's 6 nitrogen peaks and there's 3 nitrogens per tautomer. So we can tell that there are 2 tautomers present. And we're going to zoom into this area to get a better idea of which tautomers are present. So here, this chemical shift at about 250 ppm is much higher. So it represents a non-protonated nitrogen, which has a higher chemical shift. And by the carbon connectivity, we can tell that it's the tau tautomer. And these 2 green peaks, which are connected here, are a lower chemical shift, which means that they are protonated. So they correspond to the charged state. And using these labeled nitrogens and carbons, we can assign all the carbon peaks to the carbon-carbon spectrum where both axes are the full carbon spectrum. So we're going to zoom in here to get a better look. These are the zoomed in spectrums again, and you can tell that the peaks of the carbons within the same tautomer are connected. For example, the pink peaks are connected by this square, but then the green peaks of the charged tautomer are connected by the green square. So they're not connected between tautomers, but they are within. So with those techniques, we're able to identify all the carbon peaks. And we are able to confirm that there were a tau and charged tautomer present in our sample, which was at a lower pH. And this technique is applicable to other biological research. For example, the HIAPP protein, which when misfolded is causing type 2 diabetes and may be linked to Alzheimer's. And this misfolding is due to histidine tautomerism or the wrong tautomer being present. So it is important to be able to distinguish. That's all, and thank you for listening.